What's up, YouTubers? It's your favorite doctor, lawyer, teacher, plumber, astronaut, Kaibo Tang. And I myself, as a researcher, just want to present the latest paper in the field of computer vision/medical imaging. Hello. In this video, I will compare Swin UNETR and Models Genesis. So the reason why I'm comparing these models is that both models leverage pre-training. Interestingly enough, the first author of Swin UNETR is actually the mentor of a friend of mine back in high school. Anyway, without further delay, let's jump straight into the two articles. First off, the two proposed models differ in training. In Swin UNETR, the encoder maps input images to a latent space. During training, the author chooses to either directly supervise the representation in the latent space or indirectly supervise after the task-specific heads that follow the encoder. However, in Models Genesis, the author decided to simultaneously train the encoder and the decoder, and supervision is performed after the head attached to the decoder. The most significant difference between Swin UNETR and Models Genesis is that despite both models apply multiple transformations to the images before using them for training, Models Genesis use only one task, whereas Swin UNETR uses multiple tasks for different transformations. What are the transformations and the tasks, you might ask? I will explain this in details in the following slides, but to give you an intuition, I will use a very simple daily example. Suppose I have an exam to take tomorrow. The exam is about memorizing an entire scene in a certain act of a play. To prepare for this exam, I make a bunch of small quizzes on my own to test myself and help me study. For example, I might remove some sentences from the scene and try to fill in the blanks. Here, the removal of sentences from the play is the transformation, while fill in the blanks is the task. We call this kind of learning self-supervised. Using the same example, supervised learning is equivalent to, let's say, I'm doing mock tests, where the mock tests resemble the actual exam, and I correct myself after finishing the mock test by comparing my answers to the answer key. Now let's get back to the two articles. As is shown in the slides, Swin UNETR adopts different tasks for different transformations, whereas in Models Genesis, the author uses only one task, which is image restoration for all transformations. One advantage of using a single task is that it is easier to implement. In other words, only one task-specific head is required despite having 3 different transformations. However, using different heads for different transformations requires one to design a new task-specific head if one would like to implement a customized transformation. All right, without further delay, let's take a look at these transformations and tasks. First, we have the mask volume inpainting from Swin UNETR. This transformation masks out an area from the input image. The corresponding task is to restore the original image given the masked image. To put this task in mathematical terms, the author uses a mean absolute error calculated between the restored image and the ground truth image. Secondly, we have the image rotation. Very literally, this transformation rotates the image, or maybe a better term to use here is volume, about the z-axis by either 0, 90, 180, or 270 degrees. The corresponding loss is the cross-entropy loss of the predicted softmax probability of the 4 categories of rotation with respect to the ground truth probability. Lastly, in Swin UNETR, the author adopts the technique of contrastive learning. The motivation for using contrastive learning is quite straightforward. For example, if we want the encoder-decoder model to learn to distinguish between the cats and dogs, then a very reasonable approach is to make sure the representations of cats and dogs on the latent space separate as far as possible. On the other hand, for different breeds of cat, despite the difference between different breeds, we still need them to be closed on the latent space. To put this way of thinking in mathematical terms, the author uses a cosine similarity loss. Let's look at the formula here. To minimize the cosine similarity is the same as maximizing the denominator while minimizing the numerator. Taking a step forward, we are essentially trying to maximize the cosine between Vi and Vj here, while minimizing the cosine between Vi and Vk here. Note that here that Vi and Vj are the vector representations of the transformed views of the same patch in the latent space, while Vi and Vk are transformed views of different patches. Therefore, we need to make sure the angles between Vi's and Vj's are as small as possible, and the angles between Vi's and Vk's are as large as possible. Now let's move on to the transformations used by model's genesis. One transformation introduced by the author is called nonlinear intensity transformation. For this transformation, we first generate some Bezier curves using 4 control points. Then we apply these curves to the original image. In the article, the author suggests that these curves are quote-unquote smooth and monotonic. I would like to highlight here that the significance of monotonic transformations. As you can see here, we have 6 monotonic curves, which are either monotonically increasing, like example 1, 3, and 5, or monotonically decreasing, like 2, 4, and 6. One advantage of having monotonic transformation is that the mapping from input intensities to output intensities is 1-to-1. We also call this mapping a bijection. As a result, for a certain output intensity, there's one and only one input intensity that corresponds to it. To test this, we can simply draw a horizontal line on top of one of these curves, and we can observe that this horizontal line can only have one and only one intersection with the curve. Applying such a monotonic transformation allows the conservation of information before and after applying the transformation. Next, as for the second transformation, the author uses local pixel shuffling on various scales. Here, I would like to direct everyone's attention to 2 very interesting shuffling filters. Here we have the last 2 filters, with one being the shuffling within a 2x20 window, and the other one the shuffling within a 20x2 window. Applying these 2 filters to a given chest X-ray scan, one can blur the features of the spine, or more generally speaking, vertical features, while the other blurs the feature of the ribcage, or generally speaking, horizontal features. Such kinds of blur, along with the task of image restoration, encourage the model to learn features in different directions. Inpainting, which is a very self-explanatory term, refers to the transformation of masking parts of the image with blocks of different intensities. When using inpainting as the transformation, the task of image restoration turns into a task of interpolation. In simpler terms, the model is given a global information or a context and is asked to predict what an image looks like locally. Similarly, outpainting refers to the transformation of masking an area in the image and preserve the original intensities within this area, while filling the image with a constant intensity outside of this area. As for outpainting, the task of image restoration turns into what is called extrapolation. In simpler terms, the model is given some sort of local information and is asked to predict the context. To use a simple example to explain what inpainting and outpainting do, inpainting helps the model learn how to answer questions like, where is the heart and what does it look like, given a chest CT scan. And outpainting helps the model learn how to answer questions like, what does it look like around a heart? Now that we understand the transformations and the corresponding tasks used by the 2 models, I would like to give 2 comments. First, I think the design of the transformations of models genesis is more reasonable, and the author has provided ample explanations for his motivations to adopt these transformations. As far as I am concerned, these explanations and motivations are very reasonable and have clinical significance. And this is, in my humble opinion, what Swin UNETR is lacking. However, the use of contrastive learning in Swin UNETR is very reasonable too. And I think comparing to supervising after the decoder, supervising at the bottleneck appears to be more direct. But my question is, what is the clinical significance of teaching the model to recognize the different transformed views of the same patch? Or to put this question more specifically, in what task, namely segmentation, registration, classification, do we need such information? Or taking a step back, in what specific ways are such information helpful in our target tasks? I think to give a clear answer to these questions is hard, because after all, there's a very old saying in our field that is, machine learning is essentially a black box. After introducing the transformations and tasks, let's take a look at the specific architectures of the 2 models. Swin UNETR adopts Swin transformer-based UNet, whereas Models Genesis uses a traditional CNN-based UNet. In addition, for Swin UNETR, pre-training involves solely training the encoder, and decoder is trained and used only during fine-tuning and inference. However, Models Genesis train and use both encoder and decoder, regardless of whether the model is going through pre-training or fine-tuning and inference stage. Specifically during training, 2 articles uses different datasets for pre-training. Swin UNETR uses 50-50 samples from 5 CD datasets. Note that these samples are 3-dimensional. Models Genesis use 534 CT scans and 77,074 X-ray scans. It seems at the first glance that Models Genesis use a bigger dataset for training. However, this is not necessarily the case because X-ray scans are 2-dimensional. Comparing the amount of CD scans used during training, Models Genesis use a way smaller dataset compared to Swin UNETR. For testing set, Swin UNETR uses the Beyond the Cranial Vault dataset and Medical Segmentation Decathlon dataset. The model is evaluated based on the task of segmentation. Models Genesis, compared to Swin UNETR, use more dataset for testing. I won't be able to list them here, but these datasets include different modalities, including ultrasound, MRI, and CT, and also across different diseases and different organs. And the evaluation task involves both segmentation and classification. Now let's take a look at the experiments. This table here compares Swin UNETR to other models on the leaderboard of BTCV challenge. For the segmentation for almost every organ, Swin UNETR achieves a state-of-the-art performance. However, taking a closer look at the experiment, it's not hard to realize that this experiment is not actually very reasonable. Before explaining why, let's briefly go over every baseline model listed here. The first 3 setter-based models are based on transformer and pre-training is not used. ASPP used a CNN-based encoder-decoder model without pre-training. Both TransUNet and co-transformer use CNN and transformer-based UNet. Random patch uses a traditional CNN-based UNet, but adds a random patch sampling block. Partially annotated NN is a CNN-based model. However, on top of it, the author introduced partially annotated samples. NN UNet is self-configured. In other words, during training, not only is the ways of the model learned, the architecture of the model itself is also changed along the way. UNETR is written by the same author as Swin UNETR. The architecture of the 2 models are essentially the same, except for the fact that Swin UNETR uses Swin transformer instead of vision transformer. All of these models mentioned above are trained from scratch without any sort of pre-training. However, when it comes to Swin UNETR, the model is pre-trained on a huge dataset of 50/50 CT scans. This prompts me to ponder, what exactly is the reason behind the supreme performance of Swin UNETR? Is it because of the use of pre-training or the fact that Swin Transformer is superior than any other sort of building blocks of UNET? Therefore, the only conclusion I can reach from this example is that Swin UNETR is currently the state of the art. But the question I cannot answer is which exact component of Swin UNETR makes this model so good. This table shows the performance of Swin UNETR compared to the other models when evaluated on the MSD dataset. Kim et al. Uses a self-configuration method mentioned previously, and they call it Neural Architecture Search, or NAS. TransVW is from the same lab as Models Genesis, and also uses self-supervision similarly to its Models Genesis brother. C2F NAS, similar to the model proposed by Kim et al., uses NAS to configure the optimal model architecture. And then UNET, as is mentioned earlier, is self-configured as well. DINTS is also NAS-based. The results from this table confirms that Swin UNETR has better performance compared to AutoML models and compared to self-supervised learning models such as Models Genesis. The experiments regarding Models Genesis focus more on discussing the transferability of the model. This table shows the increase in performance of Models Genesis compared to training from scratch. The p-values suggest that these increases are statistically significant. In addition, this table also shows the transferability of Model Genesis. Pre-trained on 3D chest CT scans, Models Genesis is capable of fulfilling tasks in a different modality, such as the last one here, which is a task on MR scans, and even across different organs on liver and brain. In this table, the author discusses the influence of different transformations and their combinations on the performance of the model. In a few words, the table suggests that for all tasks, unified framework, which combines all proposed transformations together, is always among the top two in terms of performance, regardless of the task that the model is being evaluated on. Besides, the p-value suggests that the top two of each task have no statistically significant difference in terms of performance. Such a result shows the superiority of using a unified framework and its robustness across different target tasks. Comparing Models Genesis to training from scratch, Models Genesis can better leverage 3D data. To illustrate this point, let us take a look at this experiment. When training from scratch, using 3D models for training does not necessarily yield better performance than using 2D samples. However, when fine-tuning from pre-trained Models Genesis, using 3D samples outperforms using 2D samples. The last figure I'm presenting here does not necessarily provide a stronger conclusion compared to the table in the previous slide, but I think the point the author is trying to make here is the same. That is, Models Genesis has a very strong transferability. To bring this presentation to an end, I would like to discuss the pros and cons of Swin UNETR and Models Genesis. The author of Swin UNETR in this article suggests that Swin UNETR, quote, can benefit the transfer learning of various medical imaging analysis tasks such as classification and detection, unquote. I would like to pause here for a moment and note that in this experiment, the author only uses segmentation as the evaluation task. Therefore, I think it still remains as a question yet to be answered whether Swin UNETR can be used for other tasks. This is also the reason why I put a question mark here. The author also suggests that Swin UNETR is better than AutoML, which is proven in the experiments. As for Models Genesis, the author summarizes the advantages in 4 words. First, autodidactic. The training set does not need to be labeled and thus can save a lot of time and effort for manual manual labeling and annotation of the image. Second, eclectic. 3 different transformations are introduced in the paper and are said to be capable of encouraging the models to learn from different perspectives of different features of the image. Third, scalable. Compared to Swin UNETR, Models Genesis uses only one task for different transformations. Therefore, even if one wants to add more transformation for the purpose of learning more features, there's no need to use more task-specific head. Lastly, Models Genesis are very generic. Unlike Swin UNETR, which according to the author has limited transferability across different modalities, Models Genesis offers robust performance over multiple modalities and different tasks on different organs. And thanks for listening, and I will see you all next time.